Now that we talked and understand the different features of the different organelles, I would like to see and check this illustration again, but this time we're going to understand what we need to know about the cell organelles for the IMAT exam. So let's slowly go one by one and understand exactly what we need to know. So let's start from the nucleus. So we know the nucleus composed of two lipid bilayer. So in total we have 4 layers. So this one is 2 layers and this one is 2 layers. We know that the outer layer is an extension to the smooth endoplasmic and the rough endoplasmic reticulum. Inside the nucleus, we have the DNA genetic material, and we also have the proteins that compose and take an active part when it comes to the compaction of the DNA. So we have nucleosomes and chromatin and chromosomes. It depends on which stage we are in the DNA cycle that we are going to see in the future. We also have a very dense area inside the DNA called the nucleolus. And the nucleolus is the area that builds rRNA and is responsible to build rRNA. So we have the chromatin, and we have the nucleolus, and we have the nucleus, which is composed of 2 layers. So now we have the rough endoplasmic reticulum. Let's start from the rough endoplasmic reticulum. We said that the translation process starts from the cytosol, but then it goes and continues either in the free cytosol or on the rough endoplasmic reticulum. And proteins that are going to be translated in the rough endoplasmic reticulum are usually marked for excretion outside of the cell, or they are going to have very specific places and things to do inside the cell. So they are going to be, for example, inside lysosomes, and the proteins are going to be the hydrolytic enzymes for the lysosomes to stay inside the cell. So now that we mentioned the lysosome and the area that we have the hydrolytic enzymes from the lysosome, we can mention the lysosome itself. So lysosome is a vesicle which contains hydrolytic enzymes which function at around 5 pH, and when you have the lysosome fuses with another vesicle, it is going to form a specific vesicle that's going to basically take the enzymes that are going to break down what is in the other vesicle. So we know that if a lysosome bursts, a single one is not going to do much to the pH inside the cell. But if we have a lot of them burst, we are going to have something called either necrosis or autophagy. We are going to have the cell death. We can cause a serious damage to the cell when we change the pH. Another very important part, other than the lysosomes, the nucleus, and the smooth endoplasmic and the rough endoplasmic, is the smooth endoplasmic reticulum. The smooth endoplasmic reticulum is the area when you function and create the actual plasma membranes for different places. It's the area of detoxification, it's the area of fat synthesis and fat metabolism, which is very important. We also mentioned about the mitochondria. The mitochondria is the area where you create cellular respiration and where you create high amounts of ATP. You will see that the mitochondria contains many different lipid bilayers and proteins and structures inside of it that are going to allow us to create and yield energy from different molecules. But we are going to touch it in the future because it's quite difficult to understand and it's quite complex. For now, just remember that the mitochondria is the powerhouse of the cell. Then we mentioned the cytoskeleton. Each cell contains a cytoskeleton which is composed of microtubules, microfilaments, and intermediate filaments. The microtubules are composed of tubulin, from alpha and beta tubulin. The microfilament is composed of actin rods, and they form double helix of actin rods. And the intermediate filaments are changed between different cells, and they compose of different things, different proteins between the cells. All 3 of them are in charge of the structure of the cell, of the transportation of different things inside the cell, of the feature of the cells, of the meiosis, mitosis of the cellular when it comes to cleavage furrow and the replication, duplication of the cell. They have a lot of very important features. The next part will be the plasma membrane. The plasma membrane is a phospholipid bilayer, which we are going to talk about in the next video, which is going to border the entire cell. I didn't talk about the I didn't talk about the plasma membrane yet because I'm going to start the plasma membrane very soon, and we are going to see that we can break down the plasma membrane into many different lessons, because we have receptors and proteins and channel receptors and channel gates and channel proteins, which are going to be slowly built over the time, and you're going to see it is more complex than just a barrier between the outside and the inside of the cell. So we are going to keep it in the future. Then we talked about the centrosome. Centrosome is composed of 2 centrioles, and 2 centrioles each one composed of 9 triplets of microtubules. A very important fact to remember. And 2 centrioles are going to give us centrosome, which is also known as the MTOC, the Microtubule Organization Center. Then we talked about the peroxisomes. Peroxisomes are quite complex, but they have a lot of features. The main one would be the reduction of oxidative species and the containing of hydrogen peroxide, which we are going to discuss everything in the future. Then we mentioned that we have a vacuole inside, especially plant cell. It is going to contain some sort of a backup of water and liquids. Then we mentioned the Golgi. The station after the ER is the Golgi. And in the Golgi you do some sort of chemical modification, specific chemical modification, such as phosphorylation and the addition of carbohydrate groups and sulfurylation, adding of the sulfur group, and many different chemical additions and modifications that we are going to do to the protein that was translated inside the ER, and then with the vesicle it went to the Golgi apparatus. And we mentioned that we have a cis phase and the trans phase. The cis phase is in the same direction of the nucleus, the trans phase is in the direction of the outside of the cell. And you have many different layers for the Golgi, which is also important, and the vesicle are budding off and going to the next one, each some sort of station. And I believe that's it. We also of course mentioned the ribosomes, which are not membrane bound, and they are the area where you do the actual protein synthesis and production. And you have the ribosomes in bacteria and eukaryotic cells. In eukaryotic cells they are bigger than the bacteria cells. And this is overall what you need to remember when it comes to the IMAT and BMAT exams, when it comes to the cell biology. Now, in the future, we are going to dive into the cellular respiration. We are going to understand the plasma membrane in way more details, because these two processes are way more complicated. For example, the plasma membrane, as I said, is composed of many different proteins and phospholipid bilayer and different molecules. And the mitochondria actually can create energy, and it can recharge ADP into the form of ATP. And these two processes and structures are quite complex, therefore we are going to wait a bit and then we are going to discuss them in a few lessons. Also, don't forget, if you still didn't, to check the link in the description and to join our free course, because this is a completely non-profit and we believe in free education. But I'm doing these lessons because I want to give you the highest skill point for the IMAT and BMAT exam, especially for people who already study the material and understand the material. This is why it's so important to read the material before you actually watch the videos, which are going to be a lot of facts and very high yield facts for the IMAT and BMAT exam. I will see you next time.